Hi, everyone. Today's lecture is titled History of Vaccinology and Vaccine Science. It's going to be a longer lecture, so please feel free to pause or take a break whenever you need to. The quiz for week 1 will include content exclusively from this lecture, so make sure you just pay close attention so you're able to do well in the quiz. By the end of this lecture, you should be able to recognize the natural course of infection and how vaccines can interrupt this process, define what a vaccine is, explain the benefits of vaccination, describe herd immunity and what R naught is, distinguish the first method of vaccination and how it's utilized to eradicate smallpox, and generally be aware of how vaccines have changed over time and the future of vaccinology in the field itself. Also, you should be able to identify the steps for how vaccines are developed, approved, and manufactured. And lastly, be able to just recommend specific vaccines for US and global citizens. Okay, so I want to start off with a diagram showing the natural course of disease progression and where vaccines really have an impact. We start with this gray person here. This gray person who is susceptible to any pathogen causing a disease. Note there are varying levels of susceptibility for any pathogen just due to environmental, genetic, and host factors. And so let's say the susceptible person was exposed to a virus, for example. Now this person's exposed, and there's a certain incubation period between the time when the person was infected to the time when they start to develop symptoms. And sometimes for some infectious agents, people can be infectious for days before they even develop any symptoms. But we have this red person here that's now infectious. And so this infectious person can, of course, transmit this figurative virus to other susceptible people. But over time, there could be a variety of different clinical outcomes from this disease. And for example, the patient could have a really good immune system and fight off the virus so that he or she is recovered. However, with many infectious agents, even though you're recovered, you can still be susceptible to either that same pathogen again, or a variant of that specific pathogen. And also, just natural immunity wanes over time, which means decreases over time. So you can become susceptible again. That's why I have this gray person here, even though they're recovered. Or you could recover from the infection, but still have sequelae. And what does that mean? That means you can still experience residual symptoms from the infection, even though that pathogen has been like totally cleared from your body by your immune system. And we actually see this with SARS-CoV-2 infection, where some people are called long haulers and still can't smell or taste over a year after they've been initially infected. Or lastly, you can have the extreme case where the pathogen just becomes so deadly and the body can't fight off that infectious agent. Many times, actually, people who do die from an infectious agent, they actually die because of the immune response that's developed, otherwise known as called the cytokine storm. And you might have heard about the cytokine storm in relation to SARS-CoV-2, where again, it's that person's immune system just being overactive and killing the patient rather than the pathogen itself. So where does vaccines and vaccination come in? Let's say the susceptible person is vaccinated before they've ever been exposed to this hypothetical virus. And this is called just prophylactic vaccination or preventative vaccination. So when they are exposed, the person can become immune. Now, as you're probably aware, there are also so many different types of immunity and outcomes that can occur once you are vaccinated against a specific pathogen. No vaccine is 100% effective. And that's why I have this person right here in a lighter shade of green. So depending on the vaccine, the type of vaccine and how it works, as well as even just the patient characteristics, vaccines generally have three tiers of defense. The first tier is full protection against infection, meaning the virus or the pathogen can't even infect the person in the first place because the body is already primed to fight the specific pathogen because of the vaccine. So if the person is never infected, well, the pathogen never really gets inside their cells, then this exposed person can't transmit the pathogen to anyone else. The second tier is protection from serious illness or severe disease. And so you could potentially get infected, even though you've been vaccinated, but the vaccine prevents the pathogen from replicating too much and causing severe disease. In this second tier, this also limits the prevention of transmission of this pathogen or virus, for example. And lastly, the third tier is protection against serious illness or severe disease by itself, meaning the vaccine's good enough to prevent severe disease, but you may still be able to transmit the pathogen to other susceptible people. But also the benefits of vaccination is developing memory so that when a vaccine person is exposed to the same pathogen or a similar pathogen in the future, they're protected. And that's where you see here where this person's well. And again, I have this in an even lighter shade of green because there's so many different variables with this, right? The vaccine can still protect the person from re-challenge. But again, vaccines also wane over time too, just like natural infection does. And that's why some vaccines need boosters. We'll actually discuss more in detail in future lectures about the immunological response to vaccines. But in general, the ability of or how good the vaccine is really dependent on the type of immune response the vaccine induces, the rate of antibody decay, the type of vaccine itself, and of course, the host genetic and environmental factors. So what is a vaccine? I have a definition here, a product administered to induce an immune response for prevention and or treatment of disease. And this is the simplest definition I can create for you all. But I want you to realize a vaccine must contain information either derived from the pathogen itself or produced synthetically to induce an immune response and provide protection for people. And not every part of a pathogen is actually immunogenic, which means they can't induce immune response. So extensive research is needed to target specific antigens within a pathogen to develop an effective vaccine. And we'll actually talk more about this in a later lecture, later in this lecture, as well as in our immunology lecture. So don't get too bogged down now. So let's go over what are the benefits of vaccines. Well, they can potentially eradicate diseases. And we see that with smallpox, and we're on the verge of eradicating polio. They can control mortality and morbidity rates. This is huge. If you can prevent people from dying, as well as prevent infections from itself, then you can prevent people from experiencing the complications of these diseases. Vaccines, they save lives. This is the most recent data I can get. As of 2019, vaccines prevent 4 to 5 million deaths annually. And honestly, that number is probably a lot higher due to the COVID-19 vaccines that have been rolled out and their impact in this pandemic. Also, vaccines protect unvaccinated people by herd immunity. And we're actually going to talk more about herd immunity in the next couple slides. What else? Well, vaccines also prevent related diseases and cancers. So to give you a few examples, if you're vaccinated with the flu vaccine, well, you can also be protected against pneumonia. Um, or vaccines also prevent cancers like the human papilloma virus or HPV vaccine, which protects people against cervical cancer, or the hepatitis B vaccine or HPV vaccine that protects people against liver cancer. Also, there's actually some really good data to support that when children are vaccinated against measles, their likelihood of getting sick from other diseases significantly decreases. The mechanism is not really understood. I can give some additional examples if anyone's interested, but it's really cool that if you're immunized as a child against measles, you're protected from other diseases that might not be exactly related to measles. Otherwise, what are the other benefits? Well, vaccines can potentially induce long-term immunity. They could prevent future infections. And vaccines are cost effective compared to the indirect and direct cost of getting sick or dying from a preventable disease. Vaccines are amazing. And to think about it, if vaccines can prevent infections and diseases, especially for bacterial diseases, well, they can also prevent the development of antibiotic resistance, right? If you're not getting infected, well, you don't need antibiotics to treat that infection. And so vaccines can really help with this too. And lastly, vaccines are certainly a tool for healthcare equity. You could see this as a controversial topic, especially with the COVID-19 vaccine distribution. Definitely not equitable distribution across the United States and globally. But in general, it is seen as a healthcare tool for equity. And just note this list that I provide here for you, it's not an all-inclusive list. There are definitely many more benefits to vaccines in general. So let's talk more about herd immunity. You all probably heard of herd immunity by now, especially with the COVID-19 pandemic, hearing experts saying like, hey, we need to reach 70% vaccination rate or higher. However, just to go over it, just as it sounds, think of a herd of cattle where the strong adult cows have to protect the baby calves and the older cows from predators because they're unable to protect themselves. And this is actually the same scenario with vaccines, where the predators are the infectious pathogens and our protections from the vaccine, which protects those in our herd, in our community who can't get vaccinated. So I would like to depict herd immunity by using an example and using the same colors that I used in the past, where you have this gray individual here that's susceptible and this red individual here that's infectious. So we start with a group of susceptible people and this one person who has been infected, and we're going to use a virus for example again. Now, when nobody in the population has been vaccinated and you have all these susceptible people, well, the virus can spread from person to person to the other susceptible people, eventually infecting almost the whole community. And so you have about 21 infected people out of the 24 people in this small community. And of course, this is just an example. But let's start over. Let's say this time we have half of the community vaccinated. Now you have these green individuals who are now vaccinated. And over 50% of them are. Well, the virus can still spread and infect others in the community that are susceptible. However, with more than half the percent of the community being vaccinated, the spread of the virus is significantly reduced. So now you only have 8 people infected. So less people suffer from disease complications in general because of the vaccine. And this is how, and this in general is an example of vaccine coverage that's actually below the threshold for herd immunity, meaning the virus can still spread. And I think it's important to know, as I discussed before, vaccines can't protect every individual in the population directly. Again, no vaccine is 100% effective. And as you're aware, some individuals can't or aren't vaccinated for various reasons, either by choice, they're might be too young, either due to pregnancy, they're allergic to the vaccine itself, they could be immunocompromised, or others who just don't mount a protective immune response despite being vaccinated. And there is, of course, those who are vaccinated, those green people, but that immunity has waned over time. So that's why herd immunity is so important, because if enough individuals in the population are vaccinated, and if the vaccine prevents not only the disease, but also the infection itself, preventing the virus or pathogen from infecting people, well, the spread of that pathogen can cease and protect the community. And so this is my last example here, where we have more than 80% of the population being vaccinated. And look at here, there's still only that one person who's infected. And the reason is because this community has a threshold above a threshold of herd immunity above that the needed rate for preventing this virus from being spreading to other people. And just to say, you still do have these 3 susceptible people here. So that's why having such a high coverage vaccine rate, you're also protecting these people who can't get infected, who can't get a vaccine. And that's why herd immunity is so important. So how do we determine the threshold for herd immunity? I keep on referencing that. Essentially, to understand and answer this question, you have to know how infectious the pathogen is. And once you can understand that, well, then we can determine how much herd immunity is needed in the community. And this is measured by the R naught value, which is called the basic reproductive number for a given pathogen. And the basic reproductive number is simply the average number of people that one infected person can transmit the pathogen to if everyone in the population is susceptible. Essentially, if there's no vaccine. And so here I created a table of the most common vaccine preventable diseases. So you have the diseases here on the left, you have the R naught value right here, and then you have this threshold of immunity. And so, for example, if we're looking at measles, if one person is infected with measles, well then, and everyone in that community is susceptible, then that one person can transmit measles to 9 to 18 other people. That's why we need such a high vaccination rate or threshold for herd immunity. Essentially, the higher the R naught value, the more infectious the pathogen is, and the higher the herd immunity needs to be, and the higher the vaccine coverage needs to be. And so that's why I organized this table from highest to lowest, with the most infectious diseases up here to the lowest. Just know that these numbers are not value. They're just general estimates, and they definitely vary based on how scientists calculated the values, as well as the geographical location and the prevalence for the diseases in general. And secondly, I created this table to show you the power of vaccines. This is the most up-to-date information I can find. Here on the left, you have, again, the vaccine preventable diseases. You have the years here that correspond to the peak number of cases in the United States for each of the diseases. And then here you have 2019 data of the cases for each of these diseases. Just know, of course, you see 0 here because smallpox has been eradicated. In 2019, there has been no cases of polio, but polio has not been eradicated. I just want to emphasize that. There's still cases worldwide. And if you're a visual person like me, check out this data from the UK. Over here, they say notifications. It just means report or cases. And just look how powerful vaccines are. You have such a high number of cases here for diphtheria. And once the vaccine's introduced and people actually get the vaccine, look at how quickly those cases drop. The same with measles and the same with polio. And to be honest, these cases are probably even a lot higher than you see here. Because think about it, these are pretty old time points. And back then, they didn't really have good reporting systems like we do now, or even electronic databases. So this really shows you the power of vaccines. And as I mentioned before, vaccines are cost effective. Vaccine preventable diseases in adults cost the US around $9 billion annually. And this was reported by a study that really looked at the economic burden of these preventable diseases in 2015. And again, this is the most up-to-date information I can find. And this study was really cool because they really assessed the burden of how much it costs if for each of these pathogens if there was no vaccine. And this figure is definitely complicated. I don't want you to memorize this, but this diagram here is data from a systemic review and meta-analysis study that assessed the impact that pharmacists have on vaccination rates. And out of 36 studies that were analyzed, every single study found an increase in vaccination uptake when pharmacists were involved in the immunization process, regardless of their role. If they were an administrator, if they were an educator, or if they were the person who actually facilitated vaccination, in comparison to traditional providers without pharmacist involvement. And this is why we're teaching this course, because we are the key. Pharmacists are the key. So with that, it's probably a good time to take a break if you'd like to. But if you just want to power through with me, now we're going to go through the history of vaccinology. We're going to start with the first vaccine against smallpox. So smallpox is a highly contagious and deadly disease caused by the variola virus. The origin of smallpox is actually unknown. Oops, sorry. There are two strains of smallpox. They include the variola major and variola minor viruses. Basically, they're indistinguishable, but the variola minor is termed minor because it leads to just fewer symptoms. People have less extensive rash and scarring, and in general, there are fewer fatalities. Humans are the only reservoir for smallpox. Smallpox causes fever and a distinctive vesicle-filled skin rash. Once a person's exposed, there's about a 7 to 19-day incubation period where patients are actually not contagious and they're asymptomatic. Early stage of disease starts with a mouth or throat sore, which could then be coughed or sneezed into aerosolized droplets containing the virus. And of course, this leads to transmission of smallpox to other susceptible hosts. After the early stage, the virus can spread to the rest of the body, as seen in the picture here. Patients remain contagious until every single one of the smallpox scabs fall off. And that typically takes about 4 weeks after the onset of rash appears. And again, this is a deadly disease. The case fatality rate is estimated to be about 30%. And those who didn't die, others were either developed permanent scars or just were left blind. So it's a really debilitating disease. And it's estimated that there were about 400 million cases in the 20th century, which is crazy. The last smallpox case was in 1977, which we'll go over in a little bit. But let's talk about the smallpox vaccine. The first vaccine method was called variolation. And this method was developed in the 1600s in Asia by the Chinese and in India as well. And so what they would do is they would actually grind up the scabs from infected lesions of a infected smallpox patient into a powder. And then what some people would do is you would inhale that powder um, of that grinded up scabs, or via the intranasal route, or intradermally, where they would grind up those scabs or use pus from the lesion to scratch onto the skin of an infected in a person to protect them against smallpox. But then again, it's still smallpox, and smallpox still created a case fatality of 1 to 2% using this method. But those who did survive were immune. However, as we know, people with the Chinese and Indian methods, they would die. But in this case, we have Edward Jenner, who made the first observation where milkmaids developed cowpox lesions on their hands. And he noticed that these milkmaids who developed these lesions on their hands, of course, milking the cows, they were immune from smallpox. And so Edward Jenner was the first person to postulate that if we could use the technique of variolation with the cowpox virus, that we could potentially protect people against the variola virus that causes smallpox. And so he was the one who essentially created the first clinical trial. He inoculated a boy named James Phipps with the cowpox virus from a lesion from a milkmaid. And then he exposed James to smallpox itself, and he was protected. And Jenner actually repeated this on numerous kids after that. And the key difference, like I mentioned, is this is the cowpox virus. So these people who were variolated, if so to speak, were protected from smallpox and didn't die because it was a cowpox virus that they were using. So this led to the development of the first vaccine. Vacca is actually Latin for cow. And Louis Pasteur coined the term vaccination in honor of Jenner. And Pasteur actually defined vaccination as a preventative inoculation of all different kinds of material from an infectious person. And this led to the development of the first vaccines. And to note, these vaccines were actually used with the vaccinia virus and not the cowpox virus. They're not the same, and definitely not the same virus that Jenner first used. It's actually unknown how the switch occurred, but in general, there are 2 licensed vaccines and 1 investigational vaccine. And so there are 2 live vaccines. One's ACAM 2000 and the other one's APSV. These live vaccines are administered by a bifurcated needle, and the protection lasts about 3 to 5 years. And it's generally seen to be a 95% effective. These vaccines are actually in the strategic national stockpile in case if a smallpox disease is ever released or smallpox virus is ever released into the wild and infects anyone. We have it in our national stockpile. And typically I would go over how to administer the vaccine and go over any counseling points. But since you'll probably never vaccinate anyone using this method, I'm not going to go over it. But I did provide some administration information here and a really cool link that goes through how you administer the vaccine. Typically, you would just prick the individual 15 to 20 times with the bifurcated needle, but patients would need to actually be counseled on wound site care, and because they could actually transmit the virus. Remember, it's a live virus. For completeness sake, APSV stands for at Adventis Pasteur smallpox vaccine, and the APSV vaccine is actually the investigational vaccine that could be used in any smallpox emergency. And lastly, the other licensed vaccine is an inactive vaccine. I might butcher the name. It's Wyanos. vaccine. It's actually a replication deficient vaccine, hence inactive. And other names are Imabune and Imabenex. This vaccine is used for people who are immune compromised, who have HIV, who are pregnant, who have atopic dermatitis, cancer, transplant people, and all of the above. This vaccine is administered subcutaneously with 2 doses separated by 28 days apart. So, global eradication occurred using these vaccines. And mind you, there's no cure for smallpox. It was eradicated by vaccine prevention strategies alone. The last known case of smallpox, like I mentioned, was in Africa in 1977. The US actually stopped vaccinating people in 1973, and they also stopped vaccinating the military in 1983. And in general, there's a ban on smallpox research. There are only 2 labs in the world that officially store and handle the variola virus under the WHO supervision, and that's the CDC in Atlanta, Georgia, and the State Research Center of Virology and Biotechnology called the Vector Institute in Koltsovo, Russia. And of course, biggest thing, who declared global eradication in 1980 against smallpox? And I really like this magazine cover here. Smallpox is dead. And so with that, that starts our history of vaccinology, where you have variolation introduced from Asia into Europe with the smallpox vaccine. And then from there, you have the development of all different types of vaccines. And what's really interesting is that over time, when you look through the development of these vaccines, you can actually see and pinpoint that these vaccines were developed based off of scientific discoveries. And so, for example, the germ theory that was discovered in 1860s, right? So right here, that led to the establishment of live attenuated vaccines. And Louis Pasteur actually established a germ theory where essentially food that spoiled was because of bacteria, not spontaneous generation. And bacteria was the causative agent of infection and disease. Obviously, we know it's more than just bacteria at this point. But you'll see over time, like the development of cell culture technologies to grow viruses in the lab help to produce vaccines in vitro cell culture. Then you have advancements in molecular biology to create these protein and conjugate vaccines. And then, of course, the advancement of genomics, where now we have mRNA and nucleic acid-based vaccines. So I actually added this here just for completeness sake. So I adapted this figure. But just to go over a couple things, not listed on here is in 1946, right about here, where the first influenza vaccine was approved for the general public. In the 1960s through 70s, the measles, mumps, and rubella MMR vaccine was actually released into individual vaccines. The mumps vaccine, fun fact, was developed in 4 years and was the fastest developed vaccine before the COVID-19 vaccines were developed. In 1974, so right about here, the World Health Organization launched the expanded program on immunization, otherwise known as the EPI. And their goal was set in 1977 to reach every child in the world with vaccines for diphtheria, pertussis, tetanus, polio, measles, tuberculosis by 1990. Unfortunately, that goal still actually hasn't been reached. And then we develop over time to even create vaccines with reverse vaccinology, with the meningococcal vaccine. And what reverse vaccinology is really just a method of looking at the genetics and working backwards. So it's a method of producing a vaccine by studying the genomic information of the organism to determine which genes code for the candidate proteins that you want in a vaccine. And that's followed then by in vitro and in vivo testing in the lab, and then on animals, and then selection in clinical trials for vaccine development. Versus the earlier vaccines where you just get like the whole organism and you either kill it with formalin or you attenuate it respectively. And what's the future of vaccinology? So I really like this figure here where the authors separate different future vaccines based off of the type of bacterial vaccines, virus vaccines, parasitic vaccines, and even therapeutic vaccines. And there are even more potential vaccines that are being developed, even for gonorrhea and Zika. Not only are vaccines being developed for these specific pathogens here, but scientists are also working on different delivery forms for new vaccines. And for example, there are multiple methods of self-assembling protein nanoparticles and even immune stimulating complexes that are being developed. And we're not going to go over that stuff, but that's super complicated. But different formulations of vaccines are being introduced, and even a transdermal patch that could be used as a vaccine that has a microneedle in it containing the vaccine. And that's actually really cool because they're predicted to have improved thermostability. They're easy to deliver with minimal pain. And potentially in the future, patients can administer the transdermal patch to themselves. And again, it's a patch, so it's probably easier to dispose of. So there's a lot of cool stuff happening in this field, and this field is ever changing, which is amazing. So it's really fun to keep up with it. And even some of these therapeutic vaccines, vaccines for cancer, vaccines for allergies, like it's insane what science and discoveries are being done now to develop these vaccines in the future. And I just included this website here. The WHO has a vaccine pipeline tracker, which is updated regularly, and it's really cool to see if you're interested. So how are vaccines developed? I'm going to summarize the development process in 12 easy steps. Just know there are way more steps than these here, but I'm just going to go over it for presentation sakes. First, you start with the recognition of the disease, which you have to distinguish from other diseases. Then you identify the pathogen that's causing the disease. And if available, you establish an animal model for the disease, so you could do preclinical studies. And just note, not every pathogen can grow in an animal model, like a monkey or rat or mouse or anything. So there are some that don't have animal models. And then you would need to identify the immunologic correlate of a protection or immunity to the disease. We'll actually talk more about this in depth in our immunology lecture. Then after all that's done, you can start designing the vaccine and producing the investigational vaccines in the variety of platforms that we talked about. So many times manufacturers will have multiple candidates of all different platforms for a specific disease or pathogen. And then they go into preclinical trials, and that's only if there is an animal model that exists. And essentially the preclinical trials, they're trying to see, like, does the vaccine actually cause an immune response in that animal? And does that immune response actually protect the animal from infection or disease? And like I mentioned, there are some diseases that don't have animal models, like dengue. And so in that case, human challenge models can be used. And we'll actually talk about that in a future lecture as well. Number 7, manufacturers can then, if they have good data on their preclinical trials, apply for an IND, which is called investigational new drug application, basically to ask for permission for the FDA to do clinical trials in humans. After that, they'll evaluate the vaccine in phase 1 to 3 clinical trials. And again, we're going to discuss more about clinical trials in the future. and then apply for approval if they get good data from the clinical trials. If successful, they submit a BLA, a biologics license application. And then of course, then they'll obtain that approval. And once they obtain that regulatory approval, then they'll upscale that vaccine and distribute it and market it. And manufacturers make vaccines in lots, and each lot is tested to make sure that they're safe, pure, and potent. And something that you might not know is every single lot that's made by the manufacturer, before it's released out into the public, the FDA reviews each lot for safety and quality. And that's where we have phase 4, our post-licensure monitoring. And you have additionally advisory committees like ACIP, which we'll go over, that reviews all the data and makes recommendations on how to use the vaccine, and what populations of people and everything you use the vaccine in. While the FDA, they'll continue to inspect the manufacturing facilities to ensure that they have quality and safety for each vaccine. And this is the surveillance phase for safety and effectiveness, the phase 4. And one important thing in the phase 4 phase, where the vaccine's actually already in the public, pharmacists were already vaccinating people with the vaccine. There's a VAERS program called Vaccine Adverse Event Reporting System. And this is a really important system that's managed by the FDA and CDC to monitor adverse reactions that occur in the real world. So this can potentially pick up some side effects that you didn't see in the clinical trials, because now we're vaccinating a larger population and it's not a control environment anymore. And the VAERS system is actually really important for you to know, because pharmacists are actually expected to know how to submit a VAERS report if anything occurs when you're vaccinating people. And so again, if you're a figurative person and you like to see the whole process in one easy diagram, I found this great image here where you start with that basic research, discovery of the disease, the pathogen, you do the preclinical trials, you submit the IND, and then you go into the clinical trials in humans. And that's where you look for safety, efficacy. That's actually wrong, not effectiveness. We'll talk about the differences between efficacy and effectiveness and what that means in the future. But then after that, you'll submit that BLA that I talked about. You'll have the FDA review, and then additional reviews by the ACIP, the Advisory Committee on Immunization Practices. And lastly, phase 4, just surveillance monitoring. And so this is really a global picture of how vaccines are developed and improved. And so a summary of this, well, developing vaccines, it's a long and complicated process. It typically takes 10 plus years to develop. And then again, as you know, the exception is COVID-19 vaccines, and we'll heavily talk about the COVID-19 vaccines in a future lecture. The timeline for development and distribution of vaccines is really dependent on luck. Again, there's a lot of translation issues. Not everything that you see in preclinical studies translate into human models. Just because a vaccine's efficacious in animals, it might not translate and work in humans. And many times, most vaccines actually fail, and many drug companies spend a lot of money on vaccines that unfortunately fail in various stages of clinical trials. And also, it's really dependent on political will and the regulatory climate. And really, did the vaccine company work with FDA or regulatory bodies to develop the protocols? If so, then their approval rate is much faster. And of course, if there's other circumstances like a global pandemic, well, then you definitely have political will and a fast regulatory climate to get the vaccine out there if it's efficacious. Like I mentioned, this is the most recent data I can find. Vaccine development costs around $200 million to $1 billion per successful vaccine, which isn't crazy. And then again, just because you develop a vaccine, doesn't mean that there's barriers. Vaccines only work if they're actually used. And so there's immense barriers to vaccine distribution. Think about third world countries where it's so hard. There's no infrastructure or capacity to keep the cold chain going, or even to store vaccines. And like I mentioned in my introduction, lecture, I work with a nonprofit organization, FIMRIC, and I go and travel to medical clinics in really rural communities in third world countries. And when there's rainy season, there's no access to those communities. And so think about rural areas where they already have limited access to healthcare. And then you tack on a rainy season where all the streets are flooded and you can't even get by. There's no way you can get vaccines to the people who need it. Here in the US, I mean, I think we're all really familiar about the barriers. Many times we can't even keep to the vaccine schedules that we have for children and adults. And that's why the CDC developed a catch-up schedule. And we'll talk about vaccine schedules in the future. And as you're probably aware, on-time vaccination rates has really been exacerbated by the COVID-19 pandemic, where we really were behind in vaccinating children and people because we're all indoors and we were isolated and quarantining. And think about it, we're an industrialized country, never mind a third world country that doesn't have the resources or healthcare system or infrastructure that we have. And as you're all probably aware, in the US, there are significant racial and economic health disparities. And not to mention, there's social media, there's vaccine hesitancy, all different facets that impact vaccines distribution, acceptance, and vaccine coverage in different communities in general. And we'll talk more heavily about all these other topics in the future. And so here, I actually listed all the available vaccines in the United States. You don't have to know every single vaccine I have on here, but many of them should be familiar. I just want to point out pertussis is just the whooping cough vaccine. It's used with Tdap. That's what the P is, pertussis. And the varicella is the chickenpox vaccine. In December of 2020, the maker of the cholera vaccine, Vaxchora, stopped making it and selling it. So that vaccine is actually in limited supply or many times unavailable in the United States. So that's why I don't have it listed here. And another thing to point out, tick-borne encephalitis vaccine was actually recently approved by the FDA in August of 2021, literally 1 month before I developed this lecture. And currently they're in the scale-up process. And so that's why, excuse me, I didn't include it in this list. What are the available vaccines globally? And so I bolded a couple of them, the cholera vaccine, the malaria vaccine, that are generally used globally. And I do have a couple of them that have this asterisk near it, the cholera vaccine and the shingles vaccine, that are either limited or unavailable. And for example, the shingles vaccine or Shingrix vaccine made by GSK is currently only available in Europe, the US, Canada, Japan, and Australia. Those are the only countries. Otherwise, that ends today's lecture. So it definitely is a lot longer. I finished under an hour, but here are all the resources. And I wouldn't be able to give this lecture without really going in depth and reading all the literature and utilizing all these resources that we have. And so there's 3 slides to show you all the resources I used. Again, if you have any questions about this lecture or want to learn more about anything I talked about today, please feel free to email me. I'm happy to help. And with that, I hope you all have a great day.